Jose Delgado from Lakewood High. And Jose is going to hold forever a special record in the history of the high school interns here at Max Planck, because he's the first high school intern ever to volunteer to do a journal club for the whole institute. And for those who doesn't know what a journal club is, basically, as a scientist, we meet every couple of weeks to discuss new research from other labs that was published like in Nature, Neuron, etc. And so one person takes the job of picking this research, studying it, and then presenting it to the other scientists. And it is a task that is kind of hard, sometimes even for a postdoc. And Jose, as a high school student, did an amazing work with his journal club. So you are going to hold forever this record, and you set the bar high for the next year high school students. So, please.
Good morning, everyone. My name is Jose Delgado. I attend Lakewood High School, and I had the pleasure of working in the Taniguchi lab. And my lab involves studying the development and function of inhibitory neural circuits. And my presentation today is regarding the in situ hybridization of acetylcholine receptors in cortical chandelier cells. Now, I mentioned that my lab involves studying these inhibitory neural circuits and inhibitory neurons. And you can assume that chandelier cells are indeed inhibitory. But what do I mean by that? Well, there are two types of neurons in our nervous system. We have excitatory neurons, which increase the likelihood of action potentials occurring. Action potentials are the signals that the neurons release in order to communicate. And these neurons use specific neurotransmitters, such as glutamate. On the other hand, inhibitory neurons, they decrease the likelihood of action potentials or signals from occurring. And they do this by using neurotransmitters such as GABA. So chandelier cells are indeed inhibitory neurons. A little more information about these neurons. They are primarily located in the cortical and subcortical areas of the brain. The cortical area is the cortex, or the outermost layer. And they innervate specific neurons called pyramidal neurons. So in this image right here, we have this green structure which represents the chandelier cell. And clearly, you can see why they're called that, because they have a lot of projections and they look similar to a chandelier. And then they innervate, they connect to this pyramidal neuron. And they do this at a specific site called the axon initial segment, or the AIS. The AIS is where the action potential or the signal originates. So it's logical for an inhibitory neuron to innervate this pyramidal neuron at the place where the action potential occurs in order to prevent it from occurring in the first place. Now here, I have a real image of a chandelier cell, again, the green structure. And these red structures are pyramidal neurons. And you can see why it's so important to study these chandelier cells, because just one chandelier cell innervates dozens of pyramidal neurons. And it regulates all of these neurons and decreases the chances of them becoming overactive. Because if these neurons become overactive, this can have a lot of consequences in our neural function, such as epilepsy. Now, in order for these neurons to make these connections at the AIS, they first had to create a vast network of projections. But what drives these neurons to create this network? Well, we hypothesized that acetylcholine, which is a neurotransmitter, impacts the axonal branching of chandelier cells. Why do we hypothesize this? Well, my mentor, Dr. Andrei Steinig, did a couple of experiments. First off, it's known that these chandelier cells have specific receptors called nicotinic acetylcholine receptors. And he had the tissue containing these chandelier cells, and he left it alone with no external factors. And as you can see, at the baseline, they had about 7 projections. But when he placed it in a blocking solution that deactivates and blocks these nicotinic acetylcholine receptors, the amount of projections decreased to around 2. When he washed out this blocking solution, so it was back to normal, the amount of projections increased again. So here's the image of that. This is baseline, and you can see that there are 4 projections, while when the blocking solution was added, there was only 1. On the other hand, when this tissue with chandelier cells was placed in a nicotinic solution, nicotine being an acetylcholine amplifier, there were more projections. So clearly, acetylcholine does have an impact on the branching out of these neurons. And the only way for acetylcholine to have an impact is only if these chandelier cells have the nicotinic acetylcholine receptor. But this raises the question, which nicotinic acetylcholine receptor type is most dominant in these cells? Because there are several different types of nicotinic acetylcholine receptor, each having their own genetic sequence which determines their affinity or sensitivity to acetylcholine. So again, just to recap, chandelier cells are inhibitory neurons that innervate pyramidal neurons, and they do this by first creating a vast network of branches. And they're able to do this through acetylcholine by having receptors. But we want to determine which receptor type they have. So, of course, these receptors are very nanostructures. So how can we do this? Well, I did a method called in situ hybridization. And this method is used to molecularly detect and label specific RNA sequences. So this method involves our target RNA. So as I mentioned, each receptor type has their own specific genetic sequence, in this case being our target RNA. We also need a probe. So this green figure right here is a complementary sequence that can connect to our target RNA. Now this probe, it is a complementary sequence, it has a fluorescence to it. So when we analyze this tissue under a microscope and we see a fluorescent signal, that would mean that our probe is there, which also means that our target RNA is there. And this method involves 3 steps. First, the tissue preparation, which is fairly simple, and then we have probe preparation. And once we have the probe, or the complementary sequence, we then do the hybridization and allow them to connect. Now, before we did the in situ hybridization with the probe for a specific receptor, we first had to attain a negative control. So we did the in situ using a probe that connects to the mRNA of chandelier cells and one that connects to the mRNA of VIPs, which are proteins in our brain. So right here, this green fluorescent structure represents the chandelier cells, while these red fluorescent structures represent the VIPs, which are proteins. Now, we know that chandelier cells are in no way connected to these proteins. So when I overlap these images, I should not expect these fluorescent structures to overlap. So when I do overlap the image, as you can see, the green structures and the red structures are not overlapping, showing that they are separate entities, meaning that they are not connected. This shows that the protocol for the in situ does work, and this also provided us with our negative control. So now that we know it works, we proceeded to designing the probe for the in situ for a specific receptor. We decided to create a probe for nicotinic acetylcholine receptor beta 2. And this receptor beta 2 is one of the most dominant ones in our central nervous system. So we decided to begin with this one. In order to create a probe that attaches to the mRNA of this specific receptor, we first had to create an insert. This is a plasmid, a complete sequence of DNA. This green structure represents our insert. So this insert has the same genetic sequence as the beta 2 receptor. We then had the insert and we connected it to a backbone to create the complete sequence of DNA. So basically, we copied this insert and we pasted it onto a backbone in order to create this complete sequence of DNA and to then be able to replicate them using bacteria colonies. Once we did the bacteria colonies and we made sure that the sequence is correct, we then went on to do an in vitro transcription of the probe. Now, if you recall, this in situ hybridization requires a complementary sequence of the genetic sequence of the mRNA. But this is the same sequence. So we added a T3 promoter, which goes along this strand and creates the complementary sequence. And it stops at this enzyme right here, KPN1. So once we added the T3 promoter, it created the complementary strand, giving us our probe for the in situ hybridization. So we proceeded with the experiment, and we did the hybridization. These red fluorescent structures represent the beta-2 receptors. But what can we take away from this image? Well, we concluded that the probe that we used for this experiment does indeed detect the receptor's RNA, the receptor that we were looking for. So the probe was successful. In addition, these receptors are located in the same area as the chandelier cells, in the cortical and subcortical areas of the brain, meaning that there is a possible connection between this receptor and the chandelier cells. Now, this does not concretely prove that these two are related. So the next step in this procedure is to do an in situ hybridization using both the probe for the beta-2 receptor and a probe for the chandelier cell. But most importantly, this method allows us to understand the chandelier cells a bit more and to truly appreciate the cells a bit more. I would like to thank my mentor, Dr. Andre Sinek, for helping me throughout this process and for not getting mad whenever I did a simple mistake. Of course, Dr. Yasufumi Hayano and Tarun Kaniganti for also helping me and giving me a couple side projects. Hiroki Taniguchi as well, our lab leader, for allowing me access to the lab and for allowing me to participate in the experiments, as well as Ilaria Drago, of course, for taking a chance on me and allowing me to participate in this internship. And my fellow interns for making this experience fun and pleasurable. And of course, the architects. You guys have to agree this building is amazing. It's just a pleasure walking in here and looking at the structure. It's amazing. And of course, the Brain Trust Foundation for providing the donations to allow us to participate in this internship. Thank you.
Questions?
What are your future plans?
The question was, what are my future plans? Well, I'm planning on becoming a pediatric orthopedic surgeon. That's my main goal, but I've always had an interest for neuroscience, and I'm actually planning on majoring in neuroscience because it's just a phenomenal field. But my main goal is to become a pediatric orthopedic surgeon.
Another question? Yes.
The question was, where do I want to go to college? So my dream school is Harvard, of course, and but if that doesn't work out, of course, University of Florida, Duke, hopefully. You know, I have big aspirations, but.
So we use this video, you are going to use it for your college application, so I don't know if you want to say another school, so they don't
Yeah,
Okay, we are going to cut it out. Yes.
Sir.
You introduced the term backbone.
Yes, sir.
As a structural component. Is that a backbone, is that a cellular backbone, DNA structure? What exactly is it supporting that additional insulin?
Yes, so, let me go back to that. So, we have this sequence right here, which is our insert, which has the same genetic component as the beta-2 receptor. However, this is not enough to create a full sequence of DNA. And we needed a full sequence to be able to replicate using the bacterial colonies. So this backbone is just more DNA sequence that allows us to create a full DNA sequence. So what we did is first we had this complete plasmid, which we called blue script. And we cut it right here and right here. So now this space was empty, and then we inserted this green sequence in there. Yes, sir.
Yes.
The question was, what was my favorite thing that I learned from this project? Overall, designing the probe, I thought it was pretty fascinating because I had to design this from scratch. And this was the longest process. It took 4 weeks for us to get this. In fact, we got the probe Friday, so it was a fairly it was close. It was close. It was very close. So yeah, that was very fascinating.
Any other question? Awesome job.